Evening or no, good day. It's actually daytime, and I'm in Germany right now, so it's evening here. So thanks for inviting me. I will say something about the usage of ontologies in machine learning. So let's start and review briefly how machine learning works nowadays. So most of the machine learning that is now done, at least if huge data sets are available, is done using neural networks. So-called neural networks. So there are two types. There are the classical supervised neural networks shown on the left-hand side. And then there are now also so-called foundational models, which are obtained using unsupervised learning, but which can then also later be transformed into domain models using supervised adaptation. These models are huge. So supervised neural networks have millions to billions of parameters, and foundational models have billions of parameters. Examples of supervised DNN is, for example, Google Translate, and an example of a foundational model is GPT-3, which is now very famous. It's a language model that gets used a lot. So I will not go into the details of how these models work, but what is important to know is that they get trained with a lot of data. Here you have tuples of data where you have a vector of input and an outcome. For example, in the spam filter, this is the properties of the email. This is the outcome, spam or not. And here it's a bit different, but yields the same result if you do a training, a supervised adaptation. Now, neural networks have major weaknesses. These become apparent when they get used out of context or for the wrong purpose. So this is a 10-month-old article about the shutdown of AI by Facebook. And basically, why did they have to shut down their chatbots? Because the models that get trained in statistical learning are tied to specific context. The models are tied to specific training data, and the training data are often inadequate or only available in insufficient quantities. Each model has user-specified training hyperparameters and optimization algorithms built into it. Cases when adequate training data are available in sufficient quantities are very few. There are some, but they are few. Google Translate is a good example of this, where they are available. And we cannot obtain adequate models of the behavior of animate and inanimate complex systems in open environments. So, for example, we can never obtain enough data to train a dialogue model. Therefore, GPT-3 fails in longer dialogues. It can look good if you're lucky and select the right topic. It can look good a few iterations, but if you talk to it longer, you will see that it doesn't understand what it's saying. And that's because basically when you have a dialogue, complex systems interact. Now, but what can you do? You can alleviate some of these problems by using prior knowledge. And you can even overcome some of the problems. And this is shown here. So what's prior knowledge? So we have here prior knowledge is P of theta, and that can be used in statistics to restrict a stochastic space in such a way that models learn from the training samples drawn from this space have a higher accuracy than models that do not do. So when you use this prior knowledge, your probabilistic computation that is done by the neural network that is shown on the left-hand side becomes just better than without the prior knowledge. And it can be formulated using explicit stochastic networks, such as Bayesian networks. It can also be provided using applied ontology. And then in the computer science literature, it's called knowledge graph. This is what your whole conference, ICBO, which I haven't attended for 10 years, but I know what you're doing is about. You are building from the perspective of computer science, you are building knowledge graphs. And how can this be used? There's a very nice review article here that by one of the leaders of the knowledge graphs community, and it shows the paradigms that can be used to integrate ontologies into machine learning or stochastic learning. So so there's mapping-based data augmentation. Data augmentation works by improving sample features, using classes that are not present in the data. Mapping-based is that you basically map the input to an ontology so that you can basically reduce the space of the input, the the dimensionality of the input. And the one that is used in AlphaFold, which is the example I want to analyze today, is a sophisticated type of class feature uses. What does this mean? This means that the input and the class are coded into features that are provided by the knowledge graph. And they are then fused together and directly fed into the model. This is a fantastic approach, and this is what AlphaFold is using. So what is AlphaFold? So that's an encoder-decoder sequential deep neural network that has outperformed other approaches to protein structure prediction in the last year's CASPS P14 protein folding competition. You can see here that the performance of AlphaFold, shown in dark blue, is much better than the performance of other models. It stands really out. What's this measurement? This measurement is the C alpha root mean square deviation 95. So it's basically given the 95 coverage of the comparison of a prediction to real folding. This shows you the deviation of the C alpha atoms. So to remind you, this is an amino acid. Each amino acid has a carbon hydroxyl end and then an amino end. And in between you have the C alpha atom. And from this, then you have a rest. In alanine, that's just another hydrogen, but then a very complicated rest that can be put to the amino acid. And this measure basically is based on the C alpha distances between the prediction and the real outcome. And here you can see how very close these predictions are really to the, shown in green, are to the blue real crystal structure of the protein. It's quite amazing. Many years ago, I did my PhD in crystallography. It was such a pain. And now this is such an incredible advancement really for protein biology. How does this work? I won't go into the details because I only have 20 minutes. I just want to show a bit the principle to you and what matters for this ontology community that we are in here today. So basically, you have an input amino acid sequence. You want to get out the protein folding of it. Normally, the machinery to come from the linear input sequence that enters the endoplasmatic reticulum in eukaryotes and then ends in the Golgi apparatus in those folded proteins is a huge set of folding mechanisms that are energy dependent and that basically read the sequence and interpret it to create the folding. That's an energy dependent, highly ordered process that consumes energy. And this process needs now to be mimicked in vitro. I mean, in silico, in a machine. And how is this done? So there's the input sequence. There is then a database search for homologous sequences, which yields a multiple sequence alignment across species. So here you have, you know, birds and fish and human sequence, and they get aligned using a clustering algorithm. Here, then you have a pairing of the amino acids. We will look at it a bit more in a second. And then you also have, and that's the most important thing, a database search for homologous proteins that have already been crystallized, of which the crystal structure is known, and thus of which the structure of how these chains to get, how these molecules together form the protein chain is known. And then this is put into a neural network with some clever tricks. It's recycled several times. These are all the deep layers of the neural network. It's a gigantic matrix multiplication sequence. And basically then the core idea is to interpret protein structure prediction as a graph inference problem. And it is really an amazing work. And now we see here the three inputs once more. Magnified. So this is a protein amino acids from 1 to n. And again, so it's a triangular matrix giving you the relationships between those proteins. Then you have a homology-based sequence alignment of the sequence against other evolutionarily conserved homologous sequences. And you have the crystallography information for homology clusters. So you obtain a homology cluster here. And now you go through the protein sequences that are here and look in the CIF database. Are there any, MMCIF database, are there any proteins in this family that have already been crystallized? These are the three inputs that are used. And so then the software computes, based on these inputs, it computes the protein folding. And it is really amazing. The results are incredibly precise. So you can see here, for example, that they managed to predict the glucose 6-phosphatase, which has not been crystallized, but there are similar phosphatases. So the chloroperoxidase has been already crystallized, and so the model could now predict an active site where on which the enzyme can act, so to speak, and can bind what it metabolizes. And so this is amazing how this could be predicted without having also because the chloroperoxidase doesn't have a similar pocket. So basically it uses the overall folding information from the chloroperoxidase, but this pocket was predicted by homology to other proteins. So that's very, very impressive. But the system is not perfect. So you see here an assessment of the quality, and you see that it is limited to predicting proteins which are homologous to known structures and for which rich homology groups are available. So what you see here is an effect of the depths and the cross-chain contracts. So when the multiple sequence alignment is poor, you get, this is a measure of the quality of how well the prediction works against known crystal structures. And you see when you have a good coverage in the multiple sequence alignment, you get also good predictions, but the problem is that it drops very quickly when the depth of the multiple sequence alignment is less good. And what you also need to consider when you look at this graph is that here the comparison is restricted only to proteins in which at most 25% of the long-range contacts are between different heteromer chains. So what is a heteromer chain? For example, this is a heteromer chain, and when contacts exist between heteromer chains, then these are very long-range relationships within the protein, right? And here they are very hard to capture in this matrix. And basically, the algorithm then fails. It cannot predict proteins for which there are many long-range contacts. And that has to do with the fundamental restriction of neural networks, of sequential neural networks, because these are sequence models that have only a relatively short span of attention. And in this short span of attention, this short context, the information about the interactions needs to be encoded. And if that's not possible, the model fails. So when you have proteins which have many long-range contexts, then the model miserably fails. So it says the model uses MSAs and the accuracy decreases substantially when the median alignment depth is less than around 30 sequences. So this is one problem, that it needs a lot of known sequences that are sequenced, and the other one it needs, it cannot predict long-range interactions within proteins. But overall, it has to be acknowledged that the model really works very well. But I would like to review a bit more where the limitations come from and how such a result can be achieved and what the role of ontology is. So first of all, you have to consider that DNN design is like alchemy, right? So you have input data and architecture. They are chosen in a heuristic manner. So very often you just have to choose the data that you can get. So that's very true in protein biochemistry, where there are not so much data. I mean, the protein databases are big, but there are not so many crystallized proteins. And overall, if you compare protein data to, for example, language data, so language data in the web, there are, of course, many orders of magnitude more data than here. So you have to use heuristics to collect your data and choose them. Also, the design architecture of the network is chosen in a certain arbitrary way until you find something that can tackle the problem and yield good predictions. And that's a bit like alchemy. You have to try out things and until you find a good solution. It's a bit like in, cell biology, when you are culturing cells and are using growth factors on the cells, you're trying out things and suddenly you find something that works. It's a bit like this. AlphaFold is also like this, but it contains some design decisions which show very interesting the nature of the cognitive style of the scientific community that made AlphaFold. So the core assumption about spatial relationships between amino acids is abstracted from the biological reality. So in the biological reality of protein folding, it's a highly contextual process that depends on multiple proteins and enzymes in the endoplasmatic reticulum and the Golgi apparatus to basically modify the protein. It's like a scaffolding, like a gigantic scaffolding around the protein. You can imagine it like building a house. So many tools are used to fold it into shape, and then chemical modifications like disulfide bridges are introduced, and other modifications are done, acetylations and so on, and glucosylation. So that's a highly complicated process that is needed to achieve the folding as it is once the folding process is completed. Instead of modeling something like this, which is what traditional approaches have done, the assumption is just to model triangular relationships between the amino acid residues using basically the triangle inequality from linear algebra or calculus. So the Schwarz inequality is used. I have it in the here. So you have here triangle operations that use basically here you can see it, that use relationships according to triangle models with the proteins to model interactions. It's a very mathematical approach that has not much to do with how nature works, but to model short-term relationships between proteins, between the amino acid, it has an effect. And so this is what they use. And then they perform a lot of operations on the multiple sequence alignment representation to simplify the clustering and condense the clustering to a form that is amenable to representation in a neural network style, that is amenable to matrix or vector representation, which is totally determined by mathematical needs. So to satisfy the logic of the neural network, certain simplifications of the clustering result are performed. Nevertheless, despite this, like a Procrustes bed where the patient is basically stretched out to fit into the bed, here, nevertheless, despite this impedance between biology and mathematics, the performance on the proteins that are homologous to known structures is excellent. So this really shows that using the prior knowledge gives an excellent ability to DNN to identify regular and recurring patterns. So that is what stochastic learning is about. It's about identifying regular recurring patterns, and strong homologies in evolution means that protein motifs have been highly conserved. And if you have this plus the ontological knowledge stored in the CIF file, which is like, each CIF file is like an ontology of the protein, which you can represent as graphs. And if you use this information on a strong homology cluster, then you have detected a repeatable pattern, and then you can train the neural network to actually recognize this pattern elsewhere. That's a great achievement. So basically the DNN models the conservation of protein folding mechanisms in evolution as far as we have learned about them from crystallography experiments, but not beyond. So what is not crystallographied cannot be identified by AlphaFold. However, and that's good for your community to know, AlphaFold has such a huge success in biology and has, tackled such a huge and painful problem for the first time in a useful manner, that basically it is like an advertisement for the usage of ontologies in statistical learning. And I can already see that more and more papers now come out that also use prior knowledge in the way AlphaFold has done it. So it is an example of how important the work that is done at ICBO and other ontology communities is for the advancement of big data and data science. Thank you very much. Barry, it's your turn now.
Yes, so I'm just going to ask you a question. I was doing so. Well, it works better. People want to see your lovely face. So can you hear me, Jobst?
Yes, really well.
So I just have one question. So you say that the CIF files in protein databank serve as ontologies or as an ontology. And I think that you need to explain what you mean by that.
Yes. Can you hear me?
Yep.
Yes. Okay. So basically, first of all, you can see the collection of all CIF files as an ontology of protein folding as we know it. So because basically for the non-transmembrane proteins, we have now a quite good representation of the proteome in protein folding as identified via crystallography experiments. So altogether, all these files form an ontology which describes our knowledge about protein folding. Each file itself is also like an ontology because you can represent it as a knowledge graph in which you have, which represents the molecular relationships of the amino acids to each other that create the three-dimensional structure. And so you can show this as a directed graph. And each amino acid and its annotations form, so to speak, a node of the ontology. And so, of course, it is not an ontology like the BFO ontologies of the Obo foundry, but it is a way of showing the relationships between entities that form a system, which is the protein that works inside the cell.
Okay, so I think we want a little bit more than that. Let me give you a hint. So we have, on the one hand, ontologies like the ones that we've been talking about here, which represent universals which have instances in reality. And now I've complained for many years that UniProt or the protein ontology or the protein database are similar in the sense that they too represent universals which have instances in reality, because UniProt doesn't represent a sequence as it is in some organism. It's based upon some organism and some sequence from that organism, but it doesn't represent that instance. It's representing a universal. Now, is that what you mean?
Yes.
And nothing more.
Yeah, so SwissProt and also the CIF files are T-box like ontologies, right? So they are they contain universals. And actually, quite in a way, the comparison to universals is really good because you could argue that the protein, the crystal structure of the protein is in a way slightly idealized. Like, for example, if I have the universal of the beech tree, of course, the individuals in this category, the individuals that correspond to this universal all slightly deviate from the idealized beech tree that is my universal. I mean, idealized is the wrong word, but from the universal beech tree. Because, for example, I could now say I attach to the beech tree a nucleic acid sequence of the genome. But the individuals will all deviate slightly from this sequence, and that's the same here. So the CIF file of SwissProt is an information content. So how do you call it? Information
content entity.
Yes, information content entity of the universal protein folding of a protein, but the individually folded protein cells slightly differ from it.
Okay, anybody else want to make critical remarks? Questions? Yes.
So I have a question.
I think you probably should come forward or take your mask off or both.
I had a similar question as you, but as a follow-up. So the structure determination, experimental structure determination, is moving towards single molecular or ensembles that are not crystallized. So I wonder, will we have individuals rather than ensemble representations? So we have what you described as universals versus individuals.
Excellent question. So basically what you have are universal variants. So you can now, there are for a long time, there have been co-crystallizations of a protein, for example, with its ligand and also without the ligand. So an enzyme, for example, with a ligand or a substrate, that was the word I was struggling to find 5 minutes ago. So you may have so on the slide that I showed here, you see the substrate of glucose 6-phosphatase bound to its binding pocket, and this is achieved by co-crystallization experiments. So co-crystallization experiments yield different universals than universals without. That's the same for the iron binding pocket of hemoglobin, for example. And so what you get is more and more elaborate and differentiated universals, but you never get individuals. And that's because to create one protein crystal, you are crystallizing milligrams of protein. So huge amounts of protein get basically crystallized in the crystallization experiment. Then you do a diffraction experiment with X-ray. And from the diffraction pattern, you receive information about the protein structure that can only be obtained because only the crystal creates an interpretable diffraction picture from the diffraction of the gamma radiation that has short wavelengths and is therefore able to create the resolution you need. And so basically you always have a big mass of protein that you need to crystallize in order to obtain the universal that is in the database. So I would say from an ontological perspective that what we have in the CIFAR is always a universal, but we can get more complicated and more variances about the universal, but we won't get the individuals because we are talking of milligrams of protein. And this is in protein thought, this is huge amount.
And so the suggestion would be that if we did all of this properly using ontology tools, which is not what SwissProt or PDB do, then we would get an even more useful way of having prior knowledge. And you agree with that? Absolutely.
So basically, you can read even now in Biden's executive order for enhancing biotech, which I read a few days ago, it says that we have to improve the data so that we can get more AI and more data analytics in biochemistry to accelerate progress in biotechnology. But that, I think the major ingredient to achieve this is ontology usage. So it's just totally crucial. And even the rudimentary, so the CIF file is a great ontology. If you think of it as an ontology of the amino acids that form one protein, but many other, it's highly structured, but it was made by physicists. But that there's a lot in the Swiss protein, in the protein domain that has less intensity of ontology usage, and where a lot of work is needed to improve the data, and that will benefit then to a huge extent biological research, as this work by AlphaFold shows.
Good. Anyone want to have the last word? German or not? Yes? Yeah, come here.
Yeah, so I think you're, I can be convinced by you, but then from what you are saying, everything in our database is universe. We never collect any information about individuals. So if I'm in the sequence lab, I actually collect a sample from the experiment from a worm or a mouse, and I send it to the sequencer to generate the sequence. And I got the information, and I compare to whatever is out there in the database, and I find some new sequence information, and that can be considered a novel gene sequence I put in the database. And then some other people in the other lab who also did the experiment, and they put the sequence, they compared with mine, and they confirmed that we had the same information. So the sequence in the database or the protein information in the database, they're all universal.
Well, that's not true. So if you, that's a very good point. So let's look at the mouse proteome and mouse genetic sequences. So when you have individual, let's say, let's look at hemoglobin gene, right, in mouse. So when you have now thousands of loci of mouse hemoglobin genetic loci sequenced, they contain, of course, at the DNA level, they contain SNPs, single nucleotide polymorphisms. And if you now look at the signal, now with laboratory mice, it's even more tricky, but let's think of wild type mice. If you think of wild type mice from the wild, then you will be able to characterize an individual mouse with its SNPs. So if you have all its SNP sequence, not only the hemoglobin gene but all the SNPs, then you have certainly in the database an A-box content, so really an individual. But when you now look at for example, the average genetic locus of hemoglobin, then you have a universal. But of course, all the individual sequences that have contributed to our view of the mouse genome are individuals, are sequences from individuals. Yes, they are information content entities because they are stored in a computer and so on. But the real thing that corresponds to these sequences are data from individuals, indeed. And that's the whole point of having SNPs, and the whole science of SNPs is basically a science around individuals. So there are individuals in our biological databases, and not only universals. But in crystallography, they cannot be universals because you need millions of individual proteins to create one protein crystallography data set, right?
Good. Okay, so thank you very much, Jobst.
It was a pleasure. I regret not to be I regret that I couldn't make it to the conference. I hope I can come again another time. Thank you very much for your attention.